Welcome back to the deep dive. I want you to imagine, just for a second, that you have this massive master blueprint.
Right, like a comprehensive 1,000-page manual for building a car.
Exactly. But what if, by just choosing to ignore one single page of that manual, you don't end up with a car missing a steering wheel, you actually end up building a submarine?
Which sounds completely like magic.
It really does. But this level of radical transformation, just based on one omitted instruction, is basically what your cells are doing every single day. And today, our mission is to decode exactly how that happens.
Yeah, we're exploring a pretty groundbreaking 2026 paper from Nature Communications.
By Xiaohu and colleagues, right? It's titled The Functional Landscape of Alternative Splicing and Hematopoietic Lineage Commitment. Basically, we're looking at how researchers are using advanced AI to find these hidden microscopic switches in our DNA that dictate exactly how our blood is manufactured.
And I mean, the sheer scale of the data they synthesized to find those switches is just staggering. They didn't just look at one human cell line or, a single mouse model.
No, they went massive.
They did. They analyzed 271 distinct RNA sequencing samples. And this covered everything from fetal development to cell differentiation across 6 different vertebrate species.
So we are talking about tracing genetic blueprints from human umbilical cord blood and fetal livers all the way down to the embryonic tissues of zebrafish.
Right. They mapped this massive transcriptomic landscape to figure out, which genetic edits are just random biological noise and which ones are the fundamental master switches of life.
But before we get into all the machine learning models and the algorithms, I think we need to establish what is actually happening on the cellular factory floor. We're looking at hematopoiesis.
The process of making blood.
Right. And if you listening are familiar with the baseline biology, you know how stem cells in the bone marrow or fetal liver have to commit to specific career paths, so to speak?
Yeah, they differentiate down the erythroid lineage to become your red blood cells, or the myeloid lineage for granulocytes and macrophages.
Or the lymphoid lineage to become T and B cells. But what this paper really zooms in on is the actual mechanical switch inside the gene that commits the cell to that specific fate.
And that mechanism is alternative splicing.
Okay, let's unpack this mechanism because it's wild.
It is. Alternative splicing is basically the cell's way of maximizing its genetic real estate. Because a gene is not read as one continuous unbroken sentence, it's broken up into these coding segments called exons.
Which are separated by non-coding segments called intron.
Exactly. So you can visualize those exons as modular Lego blocks. Alternative splicing is the process where the cellular machinery selectively chooses which of those Lego blocks to snap together to construct the final messenger RNA.
Which then goes on to build the protein.
Right, and high-throughput sequencing actually shows that an incredible 95% of human multi-exon genes undergo this process.
So one single gene can manufacture a whole variety of different proteins just by mixing and matching those blocks. And the most prevalent way the cell alters the sequence is through exon skipping.
It literally just skips a block during the assembly.
Yeah, and if an exon is skipped, the final protein has a totally different physical architecture and therefore a completely different biochemical function.
And that brings us to the core scientific hurdle this paper is trying to solve. Because we know these exon skipping events happen thousands of times across the transcriptome.
But historically, taking just one single skipped exon and testing what it actually does, that requires years of really tedious wet lab work.
It's a monumental question. Out of millions of alternative splicing events happening across these lineages, which ones are just harmless typos and which ones are actively driving a stem cell to become a red blood cell instead of a T cell?
The paper highlights this dilemma brilliantly with the MEIS1 gene, MEIS1. It's a transcription factor that plays a huge role in hematopoiesis.
Right, so when the cellular factory is gearing up to make megakaryocytes, those are the precursor cells that generate blood platelets. The machinery selectively skips exon 12 of the MEIS1 gene.
And skipping exon 12 acts as this massive dynamic switch that shoves the cell toward that specific megakaryocyte fate. But if the cell skips exon 1 of that exact same gene,
absolutely nothing happens.
Nothing. It's just background noise. The protein functions normally. So as a researcher, how do you systematically find the critical exon 12s hiding in this ocean of irrelevant biological noise without spending a century in the lab?
Well, the researchers approached this by building a highly specialized machine learning model.
So to give a quick recap for you listening, we've got stem cells turning into different blood cells by snapping different gene Lego blocks together. But finding which missing blocks matter is basically a needle in a haystack problem. So they brought in an AI.
Exactly. They designed an algorithm to generate what they call a functional A score, or FAScore. They effectively trained this AI to act as a molecular detective.
And they didn't just look at the raw sequence, right?
No, they fed the AI 19 specific features for every single splicing event and organized them into 3 primary categories.
Let's do a quick structural recap of those 3 categories because understanding how the AI thinks here is crucial. Category 1 is dynamic expression,
which asks, does the splicing event change in frequency over time as the cell matures?
Right. Category 2 is evolutionary conservation. Has this precise exon skip been preserved across millions of years of evolution?
And then category 3 focuses on protein structure, meaning does skipping this exon delete a crucial physical domain of the protein, like a signal peptide or transmembrane helix?
And those structural features are highly predictive.
Very. If an exon codes for a transmembrane helix, which is the part of the protein that anchors it into the cell membrane and the cell skips it, that protein might suddenly just detach and float freely in the cytoplasm,
which drastically alters its function. So if an exon is highly conserved across multiple species, dynamically changes during a cell's developmental timeline, and alters a key functional domain, the AI flags it as highly likely to be functional. But here's where it gets really interesting. Getting the AI to accurately weigh those 19 features presented a massive bottleneck.
A huge one. If you're familiar with training any machine learning model, you know it absolutely relies on training data.
Right. Like if you want an AI to recognize a tumor on an MRI, you have to feed it thousands of scans of confirmed tumors and thousands of scans of healthy tissue.
Exactly. So in this genomic scenario, the scientists needed a positive set of proven functional splices and a negative set of proven non-functional ones.
The catch is they combed through decades of scientific literature and could only find 37 scientifically validated functional exons to serve as their positive set.
37 out of tens of thousands of possibilities.
And the situation was even more dire for the negative set.
Because scientists simply do not spend 3 years in a laboratory securing grant money to test a genetic edit, only to publish a paper declaring, hey, we removed this exon and absolutely nothing happened.
The publication bias is real, so they had 0 scientifically proven non-functional splices to use as a negative baseline, a microscopic positive set of 37, and an entirely non-existent negative set.
So how do you train an AI when you are basically flying blind?
Yeah, how do they fix that?
They deployed a sophisticated mathematical framework known as positive unlabeled, or PU, learning.
Okay, break that down for us.
Here's how it works conceptually. Imagine you have a massive data set of 10,000 unlabeled, entirely unknown splicing events. The algorithm takes the known biological traits from those 37 gold standard examples and uses them as a template.
To kind of comb through the unlabeled mass.
Yes. And they combine that with a technique called tenfold cross-validation.
Let's unpack tenfold cross-validation for anyone who hasn't worked directly in data science. It's basically a way to ensure the AI isn't just memorizing the data, right?
Correct. You randomly partition your data set into 10 equal subsets, or folds. You train your algorithm on 9 of those folds, and then test its accuracy on the 10th fold, which it has never seen before.
And then you repeat this process 10 times, rotating the test fold each time.
Exactly. By combining this rigorous cross-validation with PU learning, the model iteratively filters those 10,000 unknown events.
It slowly isolates the events that strongly resemble the 37 known positives.
And pushes the ones that share 0 traits with those positives into a negative pile. Through this iterative statistics engine, they generated a highly reliable positive set of 4,803 events.
And a reliable negative set of 5,245 events. They essentially inferred the negative training data out of thin air using statistical elimination.
It's brilliant. And once they had that clean, robust training data, they fed it into a random forest machine learning model.
And for those visualizing this, a random forest is an ensemble method. It creates a multitude of individual decision trees during training and then merges their predictions together to get a highly accurate and stable result.
And the performance metrics on this specific model were off the charts. It achieved an AUC-ROC score of BETO.993.
Let's put that score into perspective for the listener.
The AUC-ROC metric essentially grades how well a model can distinguish between 2 classes in this case, functional versus non-functional genetic switches.
Right, a score of 0.5 is equivalent to a random coin toss.
And a 1.0 represents perfect, flawless clairvoyance. So achieving a .993 means this random forest model is stunningly accurate at identifying functional splicing events from raw genomic data.
So, just for a quick checkpoint,
Yeah.
they trained this AI using just 37 examples, used stats to build a massive data set, and got a model that is 99% accurate at finding these genetic switches.
Exactly. And with that level of accuracy, they took their newly minted FAScore algorithm and just unleashed it on the human and mouse genomes.
They evaluated over 30,000 splicing events and classified them, suddenly having a prioritized, highly accurate master list of the exact genetic switches that dictate blood cell fate.
And to organize that massive master list, the researchers mapped these predicted functional splices across the evolutionary timeline. They categorized them by their evolutionary age.
And they found some splicing events are primate-specific, meaning they only exist in humans and closely related species like macaques.
But as they looked deeper into the data, they isolated a group categorized as class 5 splices.
These are ancient.
Very ancient. Genomic edits that date back roughly 429 million years. We are talking about the dawn of jawed vertebrates, long before mammals or even terrestrial life existed.
That transition from data science to paleobiology is where this paper gets truly profound. You have a computer model sorting DNA switches by how many hundreds of millions of years they've been active. What did the FAScore reveal about those ancient class 5 splices?
Well, what's fascinating here is the data showed a massive, statistically significant spike in predicted functionality for these ancient splices compared to the younger primate-specific ones.
Which reinforces a fundamental rule of evolutionary biology. If nature preserves a highly specific genetic edit, a single exon skip for 429 million years across fish, mice, and humans.
It is almost certainly load-bearing. It is performing a biochemical function that is absolutely critical for survival.
It's the biological equivalent of finding a foundational line of code in a modern cloud-based operating system that was originally written in the 1980s. If it has survived every single software update for 4 decades, you do not delete it.
Because it is probably holding the entire architecture together.
But of course, a high FAScore and an evolutionary correlation are still, ultimately, algorithmic predictions. To prove that these specific exon skips actually function as master switches in hematopoiesis, they had to take the AI's predictions and validate them in living organisms.
Right, time for the wet lab validation.
So, recap time again. We know the body makes blood by skipping exons. The AI found the important ones and showed that the oldest ones are the most critical. Now they're going to test if messing with those ancient switches actually crashes the system.
To do this, they selected 4 top-scoring candidates from the AI's hit list to test in mouse stem cells: KLF6, SSBP3, EPB41L1, and PBC1D23.
The first 3 are heavily involved in regulating gene expression and cellular structure. KLF6 is a well-known transcription factor. SSBP3 binds to DNA to regulate transcription.
And EPB41L1 is involved in the cytoskeleton. But the researchers ultimately focused their deepest validation efforts on the 4th candidate, TBC1D23.
Let's focus on the start of the show. Why TBC1D23, and specifically exon 15 of that gene?
Because the AI assigned exon 15 of the TBC1D23 gene a near-perfect FAScore of 0.998. And it did so specifically within the human erythroid lineage, which is the developmental pathway that generates red blood cells.
So to test if the AI was right, they used CRISPR-Cas9 gene editing technology. You can think of CRISPR as highly programmable microscopic scissors.
They designed guide RNAs to direct these scissors to cut out just that tiny exon 15 from the DNA of living mice while leaving the rest of the vast TBC1D23 gene perfectly intact.
They deleted a single paragraph from a massive blueprint. What actually happened to the physiology of those mice?
The physiological impact was severe. The mice suffered from profound anemia. The laboratory observed a massive drop in total red blood cell counts.
Wow.
There was a corresponding drop in hematocrit, which is the volume percentage of red blood cells in the blood, as well as significantly depressed hemoglobin levels.
But what about their white blood cells?
Their white blood cell counts remained completely normal.
That precision is terrifying.
It proved that deleting this single microscopic exon selectively crippled the red blood cell factory, leaving the immune cells untouched, exactly as the FAScore AI predicted.
But they didn't stop with the mouse model. Because this exon is an ancient class 5 splice that evolved hundreds of millions of years ago, they went back down the evolutionary tree and tested it in zebrafish.
Yeah.
How do you test an exon skip in a fish embryo?
In the zebrafish model, they used morpholino treatments.
Morpholinos, okay.
They're essentially synthetic oligomers, custom-designed molecules that bind to a specific sequence of target RNA. By binding to the RNA, they sterically block the cell's splicing machinery from accessing that exon.
It just physically gets in the way and forces the cell to skip it.
Exactly. And when they suppress the inclusion of exon 15 in developing zebrafish embryos, the results mirrored the mice. At 48 hours post-fertilization, they recorded a 45% reduction in red blood cells.
And an almost 50% drop in hemoglobin levels. They actually visualize this using a specific chemical stain that turns hemoglobin dark blue, making the stark lack of blood cells highly visible under a microscope.
So across vast evolutionary divides from a mammal down to a teleost fish, that one single exon is a load-bearing pillar for manufacturing red blood cells.
Which brings us to the ultimate how and why question for you to consider. How does missing one microscopic fragment of a gene cascade into systemic anemia across entirely different species?
To understand the molecular butterfly effect here, we have to look at the resulting protein cascade. That tiny exon is only 45 base pairs long.
It codes for exactly 15 amino acids. It is a minuscule fraction of the overall TBC1D23 protein structure.
But those specific 15 amino acids act as a crucial docking station. They dictate how the protein physically interacts with other molecular machinery in the cell.
And the researchers discovered that when exon 15 is skipped, the protein shape changes in a way that dramatically elevates its capacity to bind to a nuclear pore complex protein called RNBP2.
The shape changes, and it suddenly binds much tighter to RNAP2.
What does that anomalous binding event trigger downstream?
That tightly bound complex initiates a destructive molecular domino effect. It significantly increases a specific chemical modification called SUMOylation.
SUMO proteins are small tags that get attached to other proteins to alter their function, right?
Yes. In this case, the hypersummoylation targets another crucial protein called HDAC1.
And for context, HDAC1, or the histone deacetylase 1, is a major epigenetic regulator. It removes acetyl groups from DNA, which causes the DNA to wrap tighter around histones, effectively silencing or repressing transcription.
Precisely the right context. When HDAC1 becomes hypersummoylated due to this missing exon, its repressive activity goes into absolute overdrive.
It just shuts everything down.
It alters the expression landscape of thousands of downstream genes. It effectively hits the emergency brakes on the entire erythroid developmental pathway, halting the production of mature red blood cells at the epigenetic level.
That is an unbelievable chain reaction. Think about it, deleting just 15 letters of amino acid code changes a physical docking site. That triggers an abnormal chemical tag on an epigenetic regulator, which wraps the DNA too tight, shutting down thousands of downstream genes.
Crashing your red blood cell count and leaving the organism anemic.
And the AI predicted this entire critical bottleneck just by analyzing dynamic expression and evolutionary conservation data.
It is a profound validation of the FAScore algorithm. Uncovering that exact biochemical pathway using traditional wet lab methods would have taken teams of researchers years of trial and error.
The AI pinpointed the exact vulnerability in the armor by synthesizing data patterns that human researchers simply couldn't process at scale.
Which brings us back to you, the listener. Why does this specific methodology matter to your understanding of biology and the future of medicine?
First, it is a stunning reminder that your body is an incredibly delicate, beautifully balanced ecosystem where a single cellular typo can crash the entire system.
But practically speaking, this FAScore AI isn't just a bespoke tool for blood cells. This exact same computational paradigm can now be deployed across the entire biological spectrum.
We could use it to map the alternative splicing events that drive early brain development, the splicing errors that accumulate during the aging process, or the hidden genetic switches that trigger complex diseases like cancer or neurodegeneration.
The clinical implications are massive. For decades, the field of gene therapy has struggled with the sheer volume of potential targets. We knew the human genome was vast, but we didn't know which switches actually controlled the machinery.
Tools like the FAScore allow us to systematically cut through the noise.
If we can accurately map the functional landscape of alternative splicing, we can design highly targeted therapies, perhaps utilizing CRISPR or synthetic morpholinos that repair or manipulate these specific exons.
We are looking at the potential to cure diseases at the foundational level of RNA splicing. We're essentially being handed the master control panel for cellular development.
And that leaves us with a pretty provocative thought for you to mull over as we wrap up.
Yeah, think about this. If our machine learning models can now accurately predict the function of ancient genetic edits that have been hiding quietly in our DNA for over 400 million years, Yeah. What happens when we point this AI at the vast stretches of the genome we used to dismiss as junk DNA?
We called it junk merely because we didn't have the tools to understand its function.
Are there dormant evolutionary traits, ancient biological pathways for tissue regeneration, or extreme disease resilience just sitting in our code waiting for the right exon to be skipped?
It is arguably the most thrilling frontier in modern genomics. The deeper our algorithms learn to read the blueprint, the more we realize how profoundly we've underestimated the complexity and potential of our own biology.
It completely changes how you look at every cell in your body. That is all the time we have for today. Thank you so much for joining us on this deep dive into the hidden switches of our DNA. Until next time, keep exploring and stay insanely curious.